Good morning, my dear students. In this session, we are discussing what is BT, its mode of action, and symptoms associated with the BT. So, what is the BT? Bacillus thuringiensis. It is a rod-shaped, gram-positive bacterium. It's found naturally in the soil throughout the world. This Bt bacterium produces spores as well as crystals simultaneously but separately. When this bacterial cell dies, it releases both the spores and crystal toxins into the environment. The insect ingests both the spores and the crystal proteins from the environment while they are feeding. These crystal proteins are the actual insect killing toxins which dissolve in the insect gut, activating its toxic effect. The spores germinate inside the insect gut, allowing Bt to multiply and continue its life cycle. Different strains of Bt. Different strains of Bt refers to the variations or subspecies of the bacterium Bacillus thuringiensis. Each strain produces a distinct set of insecticidal proteins known as cryproteins or delta endotoxins. These cryproteins are toxic to specific group of insects when they ingest and have been utilized extensively in the development of genetically modified crops to confer resistance against certain insect pests. For example, strain that is Bt Kurstaki, it is effective against Lepidopteran pest, Bt israelensis and Bt sphericus. It is effective against dipteran pest, Bt isy. It is effective against Lepidopteran pest, Bt japonensis effective against Lepidopteran pest, Bt variety tenebrionis effective against Coleopteran pest, Bacillus papillae effective against Japanese beetle larvae. See here the mode of action of the Bt. Steps by steps process. First, ingestion of the Bt crystals and spores. Insect consumes Bt spores and crystals from the sprayed plant or transgenic crops. This Bt crystals contains a 130 kilodalton protaxin which remain inactive until it is processed in the insect midgut. Next, activation of the toxin. In the alkaline pH of the midgut, the Bt crystal dissolve and the protoxin is get activated into a 60 to 70 kilodalton active toxins by the action of protease enzyme in the midgut of the insect. These toxins upon activation penetrate the peritrophic membrane and bind to the specific receptors which are present on the midgut epithelial cells. This activated toxin cause gut destruction. This activated toxin cause gut destruction by forming pores into the gut cells. Due to the formation of the pores in the gut cell leads to cell rupture, ionic leakage into the hemolymph, and gut failure that prevents insect feeding. This damaged gut wall allows the spores to enter into the body cavity, that is into the blood or hemocoel of the insect, where the spore germinate into bacteria, and this bacteria multiply rapidly in the hemolymph, and this growing bacteria release toxins and digestive enzyme disrupting insect body functions, and the insect dies from septicemia, that is blood poisoning, within 2 to 3 days. See here clear picture. Ingestion of the Bt crystals and spores. This crystal dissolve in the insect midgut alkaline pH, and the passive toxin is get into active toxin of 60 to 70 kilodalton. This activated toxin bind to the specific receptors which are located on the midgut epithelial cells, and they form pores. Due to the formation of the pores, the gut content leak into the hemolymph cause blood poison, that is septicemia, leads to the death of the insect within 2 to 3 days. And through these spores, the spores enter into the hemolymph, and where they germinate and grow very fastly as the bacterial cells. These bacterial cells also release toxic substances and cause the death of the insect. Then what are the symptoms associated with the Bt action? What are the symptoms associated with the Bt toxification? In Bt toxicated insect, the head capsule become large. The head capsule become large compared to its body size. Paralysis. The Bt proteins damage the insect gut lining, leading to the paralysis. There is no movement. Starvation. The insect stops eating and dies from the starvation. The insect dies within few hours to week. After death, the larva turn black in color. After death, the larva gives off a foul smell. So, this is all about what is the Bt, its mode of action, and symptoms associated with it. Now we are discussing the objective questions which are related with this topic. What type of bacterium is Bacillus thuringiensis? Gram negative, gram positive, acid fast, or none of the above? So, Bacillus thuringiensis is a gram positive bacterium. Where is Bt naturally found? Deep oceans, soil, human gut, air. Here correct answer is B, that is soil. Bt naturally found in the soil everywhere in the world. What structure does Bt produce that kills insect? Exotoxin, cryproteins, endotoxin, lipopolysaccharide. Here correct answer is the cry proteins. Bt produces cry proteins that kills the insects. What does the term delta in delta endotoxin refers? The acidic nature of the toxin, the crystalline shape of the toxin, the DNA structure of the bacteria, the presence of endospore. Here correct answer is the crystalline shape of the toxin. The term delta in delta endotoxin refers to the crystalline shape of the toxin. How do insect ingest Bt toxin? Through inhalation, by direct injection, by feeding on Bt spores and crystals, by absorbing through their skin. Here correct answer is by feeding on Bt spores and crystals. Insect ingest Bt toxin by feeding on Bt spores and crystals. Where does the activation of the Bt toxin occurs? In the insect hemolymph, in the insect gut, in the insect nervous system, outside of the insect body. Here correct answer is the in the insect midgut, that too in the alkaline pH. Activation of the Bt toxins occur. What pH condition is required to activate Bt toxin in the insect midgut? Acidic pH, neutral pH, alkaline pH, or none of the above? Here correct answer is alkaline pH is required to activate Bt toxin in the insect gut. What happens when the Bt toxin binds to gut receptors? It increases gut motility, it forms pores in the gut cells, it dissolves completely, it excreted as waste. So here correct answer is B, that is it forms pores in the gut cells. When the Bt toxin binds to the gut receptor, it forms pores in the gut cells. What ultimately causes insect death after Bt infection? Neurological paralysis, blood poisoning, dehydration, high fever. Here correct answer is blood poisoning, that is septicemia. Septicemia ultimately causes insect death after Bt infection. Bt Kurstaki is most effective strain against mosquito larvae, lepidopteran pests, beetles, and aphids. Here correct answer is lepidopteran pests. Bt Kurstaki is most effective against lepidopteran pests. Bt israelensis is effective against caterpillars, mosquito larvae, termites, whiteflies. Here correct answer is mosquitoes, mosquito larvae. So Bt israelensis is effective against mosquito larvae. Bt ISY is primarily targets Lepidopteran pest, Dipteran pest, Coleopteran pest, or nematode. Bt ISY primarily targets Dipteran pests. Bacillus thuringiensis variety Tenebrionis is effective against mosquito larvae, beetles, ants, termites. Correct answer is beetles. Bacillus populae is specifically effective against armyworm, Japanese beetle larvae, thrips, cockroaches. Bacillus papillae is specifically effective against Japanese beetle larvae. What is the first step in the BT mode of action? Septicemia, gut destruction, ingestion of BT crystals and spores, spore germination. Here correct answer is ingestion of BT crystals and spores. It is the first step in the BT mode of action. What is the weight of the protaxin protein form of cryprotein? What is the weight of the protoxin form of the cryprotein? That is 60 to 70 kilodalton, 130 kilodalton, 200 kilodalton, 50 kilodalton. Correct answer is 130 kilodalton. What is the weight of the activated toxin after gut proteases cleaves the protoxin? So correct answer is 60 to 70 kilodalton. What happens to the insect's gut when Bt toxin acts? It becomes more elastic. It forms pores causing cell lysis. It contracts rapidly. It digests food more efficiently. Here correct answer is it forms pores and cause cell lysis. What symptom is observed an insect that dies from BT? Greenish body color, head capsule enlarges, increased appetite, transparent body. Head capsule is enlarges than their body. After the insect dies, what color does its body turns? Red, black, white, yellow. It turns into the black color. BT in agriculture and GM crops, that is, here question is which genetically modified crop contains BT genes for pest resistance? BT wheat, BT corn, BT tomato, and BT soybean. Correct answer is BT cotton. BT cotton contains BT gene for pest resistance. What is the main benefit of using BT in genetically modified crops? Increased plant growth, reduced use of chemical pesticides, increased soil fertility, improved water absorption. Here correct answer is reduced use of chemical pesticides. So this is the main benefit of using BT in genetically modified crops. Thank you. That's it from my side.
Good morning, my dear students. Myself, Dr. Rathod Lal Singh, Assistant Professor in the Department of Agricultural Entomology at School of Agriculture, SR University. In this session, we are going to discuss about one of the most important biopesticide, that is EPN, entomopathogenic nematodes. Entomopathogenic nematodes, these are the soil inhabiting nematodes or organisms. This nematode causes diseases in insects. That's why we are calling them as entomopathogens. This nematode lives inside the host body. That's why they are considered as endoparasitic or endoparasitic nematodes. The nematodes, which belong to the Sternernemachidae family and Heterorhabditidae family, are most useful as the biocontrol agent in the various insect pest management. We will see the different species and host insects in coming slides. Entomopathogenic nematode has 6 life stages. They are juvenile 1, juvenile 2, juvenile 3, juvenile 4, juvenile 5, and juvenile 6. In general, that is, in case of plant parasitic nematodes, juvenile 2 stage is infective or effective 1, but in case of entomopathogenic nematodes, juvenile 3 stage, that is J3 stage, is infective or effective 1. This entomopathogenic nematode have symbiotic relationship with the bacteria, and this symbiotic relationship is compulsory in order to kill insect pest. If there is no symbiotic relationship with bacterium, then these nematodes are ineffective or useless. See here the example for nematode bacterium complex. Sternernema nematode, which is associated with the Xenorhabdus bacteria. Heterorhabditis nematode, which is associated with the Photorhabdus bacteria. Then, what is the benefit for nematode from this symbiotic association from the bacteria? Bacteria kills the insect host, create a suitable environment, break down the host tissues, and provide the food source for the nematode. These are the benefit to the nematode from the bacteria. Then what is the benefit for the bacterium from the nematode in this symbiotic association? This nematode helps the bacteria in the penetration of this bacteria into the host hemocell, and this nematode protect the bacteria from the external environment as well as from the host antibacterial proteins. Otherwise, this antibacterial protein neutralize that bacteria. Like that, in this symbiotic association, bacterium is benefited from nematode and nematode is benefited from the bacteria. See here the scientific classification of Sternernema nematode. It is belong to the kingdom Animalia, phylum Nematoda, class Chromadoria, order Rhabditida, family Sternernemachidae, genus Sternernema. See the various species under Sternernema nematode and their host. Sternernema carpocapsae. Sternernema carpocapsae, it is effective against corn earworm, that is Helicoverpa zea, fall armyworm, codling moth, European corn borer, and various beetles and other soil-dwelling insects. Next species is Sternernema feltiae, which is effective against fungus gnat, root weevil, cutworms, carrot weevils, and various soil-dwelling pests. Next species is Sternernema glazeri. It is effective against sugar beet root maggot, wireworms, corn rootworm larvae, various beetle larvae, and other soil-dwelling insects. Next species is Stenarnema crassi. It is effective against weevils, fungus gnat, and various soil-dwelling insect pests. Next species is Stenarnema longicauda, which is effective against wireworm, beetle larvae, and various soil-dwelling insects. Next, see the scientific classification of Heterorhabditis nematode. This Heterorhabditis nematode belongs to the kingdom Animalia, phylum Nematoda, class Chromadoria, order Rhabdicida, family Heterorhabditidae, and genus Heterorhabditis. See the species under Heterorhabditis nematode. Heterorhabditis bacteriophora. It is effective against corn or rootworm larvae, beetle larvae, weevils, cutworms, and various soil-dwelling pests. Heterorhabditis indica, it is effective against sugarcane borer, root weevils, fungus gnats, various soil-dwelling insects. Heterorhabditis marillatus, it is effective against root weevils, beetle larvae, wireworms, various soil-dwelling pests. Next, Heterorhabditis downsea. Heterorhabditis downsea, it is effective against beetle larvae, cutworms, various soil-dwelling insects. Heterorhabditis safricana, it is effective against root weevils, various beetle larvae, and soil-dwelling insects. Next, we are going to see about mode of action of entomopathogenic nematode in brief. This entomopathogenic nematode actively forays and seek the host in the soil. Once it find its host, it penetrate into it through the natural openings such as anus, mouth, spiracles, or any other sharp part of the body, and cause infection by releasing bacteria. Inside this host, the bacterial pathogen release toxins and other toxic enzymes that cause pathogenesis, that cause diseases in the host insect and eventually kills them. On this dead and decaying host, the nematode feed, grows, reproduce, and produce progeny, and this progeny will disperse for attack to the next new host. See here in detail the mode of action of entomopathogenic nematodes, foraging and host seeking. Entomopathogenic nematodes actively search for the insect host in the soil. They are attracted to the chemical signals such as carbon dioxide and other compound emitted by their target host. These chemicals guides these nematodes towards their potential host. Next step in the mode of action is the penetration and infection. Upon locating a host, entomopathogenic nematode enter into the insect body or host body through the natural openings like mouth, spiracles, or soft areas of the cuticle. Once inside, they release symbiotic bacteria, either Xenorhabdus or Photorhabdus. It depends on the nematode species. Bacterial pathogenesis. The bacteria inside the host develop and multiply rapidly, secrete toxic enzymes and toxic material that break down the host tissues and suppress host immune system, leading to the death of the host. Nematode feeding, reproduction, and dispersal. Entomopathogenic nematodes feed on both the bacteria and the decaying host tissues. It feeds on the bacteria to carry its infectious nature, infectious nature to attack the next host. And decaying host tissue, it feeds for its development and reproduction. As these nematodes consume these sources, they grow and reproduce. The nematode produce new infective juveniles which exit the dead host and search for the new targets. Different formulation of entomopathogenic nematodes. Entomopathogenic nematode available in granular form, liquid suspension, wettable powder form, pellet form, and capsule form. In granular form, entomopathogenic nematode is mixed with a granular carrier materials. In liquid suspension, entomopathogenic nematode suspended in water or any other liquids. In wettable powder form, EPNs formulated as a powder. It's to be mixed with the water. In pellet form, EPNs are produced in pellet or crumble form. In capsulated form, EPNs are developed into a capsule, that is biodegradable capsule for slow release of the nematodes. Granular form is applied as are applied for soil dwelling pest. Liquid suspension, it is suitable for soil and foliar application. A wettable powder also suitable for soil and foliar application. Pellet form, it is also useful for plant and soil application. Encapsulated form also useful for plant, that is whole application and soil application. See here the typical dose, it is ranges from 1 to 5 billion nematode per acre, and it will be vary with nematode to nematode species and their age and stage. See here commercial formulation given here. Not grub stake high, horticultural scan mask, Nemasealed nemasys and their doses for hectare is given. That is 25 million infective juveniles can be cover 1 to 2 hectare, or we have to mix in 200 to 500 liter of water to spray 1 hectare area. See here one experimental example is given for the management of root grub in erica nut. For management of root grub in erica nut, we have to apply Steinernema carpocapsae in a liquid suspension at a rate of 1.5 billion infective juveniles per hectare. The first applications should be made during June-July with the onset of the monsoon. The second application should be done during September-October. Along with the application of this entomopathogenic nematode, apply imidacloprid. Imidacloprid 17.8 SL at 0.5 ml per 2 liter of water per farm. It acts as synergistic with the entomopathogenic nematode and enhances the control of root grubs. One important noting point: during nematode application, regularly irrigate the field to maintain optimum soil moisture. Why? Because it is a very must or which is crucial for survival, effective establishment of nematode and their movement from one place to the another place. Environmental persistence. In absence of host, that is outside of host infection, EPN do not undergo dies. Instead of this, EPN can survive the soil by entering a dormant state. They withstand various environmental conditions and remain viable, ready to infect a new host when the condition becomes favorable for them. See here the eco-friendly powder form entomopathogenic nematode formulation. It is recommended at the rate of 1 into 10 to the power of 8 infective juveniles per acre, that is 2 kg powder formulation per acre. This is the gel formulation of entomopathogenic nematode. In the final slide, what are the symptoms of entomopathogenic nematode in host insects? The symptoms include cessation of feeding, increased hemolymph pH, vomiting, diarrhea, sluggishness in host insect. We also observe we can also observe general paralysis and septicemia, that is blood poison, occur in 24 to 48 hours, resulting in the death of the insect. In particular to the Steinernema nematode infected larvae, they turn creamy or dark brown in color. At the same time, Heterorhabditis nematode infected larvae, they turn reddish or purplish in color, and they also shows sticky nature of the cadaver. Thank you. In the next session, we are going to discuss about mass multiplication of entomopathogenic nematodes. Good morning. Welcome you all. Now we are going to discuss about steps by steps process for field isolation, recovery or extraction, mass production and field application of entomopathogenic nematodes. The first step is field isolation of entomopathogenic nematodes. So for this, gather soil from the different part of the field and place this collected soil into the plastic container. Into this plastic container, introduce 3 to 5 wax worm as the bait, since they are highly susceptible to entomopathogenic nematodes. Then after, close and keep the container in the dark room at 20 to 25 degrees Celsius and check the worms for the sign of EPN infection over 3 days. The infected waxworm will change their color and they die. If no infection is observed, collect the new soil sample and repeat the same process until the infection is detected. If there is infection, note the color change in the infected waxworm to identify the EPN species. For example, if the waxworm is infected with Heterorhabditis bacteriophora, it develops brick red or purple color. If it is attacked by Heterorhabditis migidis, it develops orange color. If it is attacked by Steinernema glazeri, it develops cream or brown color. So like that we can isolate EPN from the field. Next, recovery of entomopathogenic nematodes from the infected cadavers of the waxworm by using white traps. So for this, first rinse the dead cadaver of the waxworm with the sterile water to clean off soil and dirt. Next, cut a Wattman filter paper to fit a convex watch glass and place this watch glass in the petri dish. Moisten the filter paper with the sterile water and place it over the convex watch glass which is already placed in the large petri dish plate. Next, place the cadaver and the filter paper with the little distance between them for easy movement of emerging juveniles. In this Petri plate, pour 20 ml of sterile water to keep the filter paper moist. Over 7 days or over 7 to 10 days, EPN will move into the water and gives the appearance of cloudy. The water will give the appearance of cloudiness. It indicates the nematode emerged from the waxworm larvae. Like that, we can recover entomopathogenic nematode from the cadavers of the waxworm. Now, we can use this cloudy water, cloudy entomopathogenic infected water for the mass multiplication of the entomopathogenic nematode. See in the next slide, mass production of entomopathogenic nematodes. Pour 2 tablespoons of EPN infested water over 250 waxworm larvae in the container. So this container contains 250 waxworm larvae over which we have to pour 2 tablespoons of entomopathogenic nematode infested water. Keep this container in a dark, moist environment at room temperature. After 2 weeks, EPN will emerge. Yellow moss on the lid and wall of the container will indicate the emergence of entomopathogenic nematodes. Now collect them by sieving the content and washing with the distilled water. So we can collect in this tub or container. Next, field application of entomopathogenic nematodes. Mix this entomopathogenic water solution in the spray tank and spray entomopathogenic nematode on foliage or the soil late in the afternoon or around the sunset to avoid UV damage to the nematode. Take 2 points in consideration, that is, keep the soil moist to help entomopathogenic nematode establishments and their movement. Second one is till or move the soil for easy spread of the APNs throughout the field. So, see here, each waxworm larvae can bear approximately 1 lakh entomopathogenic nematode. Therefore, 250 waxworm can bear 25 million entomopathogenic nematodes. But we need 100 to 300 million EPNs per acre. Therefore, 4 cups of EPN infested water, each containing 250 waxworms, are needed to apply the recommended dose of 100 million EPNs for the acres. Thank you. Now we will see objective type questions related with the entomopathogenic nematode. The insecticidal property of nematode was discovered by Dutke and Howe. The infective juvenile nematode is, which instar is infective juvenile of the nematode, that is third instar is infective juvenile. Thinner Nematidae and Heterorhabdidae are associated with the bacteria, whereas nematode belong to the Myrmidhidae are the obligate parasites. They do not depend on the bacteria for killing the host. They kill they themselves. In Stenor nematodes and heterorhabditids, bacteria are stored in the intestinal lumen of the nematode, and infestation result in the formation of bacterial soup or bacteremia. Bacterial soup refers to the condition within an infected host where the accumulation of bacteria and host tissue fluid create a thick, viscous mixture. Myrmidhids cause morphological abnormalities and sterility. An example is hexamermis nematode. See, this is the hexamermis nematode. Steinernema neoplectana or Steinernema glacier is the first entomopathogenic nematode applied in the field to the turf against the Japanese beetle Popilla japonica. Steinernema glacier is the first nematode applied in the field to the turf against Japanese beetle Popilla japonica. DD-136 formulation is the mixture of nematode Steinernema carpocapsae and the bacteria Achromobacter nematophilus. So DD formulation is the mixture of nematode Steinernema carpocapsae and the bacteria Achromobacter nematophilus, and it is applied as an aqueous spray for the control of codling moth, that is Cydia pomonella. This is the Cydia pomonella. For this pest management, we can use DD-136. Steinernema nematode release Xenorhabdus bacteria and Heterorhabditis nematode release Photorhabdus bacteria. Therefore, this nematode act like a small syringe to inject the bacteria into the host. In Stener nematidae, the symbiotic bacteria is from the genus Xenorhabdus, specifically Xenorhabdus nematophilus and Xenorhabdus bovini, but this bacteria do not flourish. But in case of Heterorhabditidae, the symbiotic bacteria is from the genus Photorhabdus, specifically Photorhabdus luminescens. This bacteria fluoresces glow in the dark, making the infected larvae easy to detect. Steinernema-nematid nematode infected larvae turn into the creamy or dark brown in color, whereas heterorhabditid nematode infected larvae turn reddish or purplish in color. See here the nematode and their association with the pest. Tetradonema pelicans. It is effective against Sciarid flies, that is fungus gnat, that is the pest of the cultivable mushroom. Second nematode, Romanomermis culicivorax. Romanomermis culicivorax is effective against mosquitoes. Steinernema feltiae, it is effective against soil pests and termites. Rhabditis species, it is effective against Holotricha serrata, a type of beetle pest. Based on their searching strategies, Entomopathogenic nematodes are two types, that is ambusers and cruisers. Ambusers, they remain near the soil surface and wait for the mobile host to come close. But cruisers, they actively search for the host within the soil and are effective against less motile insect. Example for ambuser is Tinernema carpocapsae. Example for cruisers is Tinernema glaceri, Heterorhabditis bacteriophora. Ambusers target soil-dwelling and surface pests; cruisers target less motile soil insects. Based on application and targeting strategies, entomopathogenic nematodes are green commandos and sea soil commandos. Green commandos entomopathogenic nematode used for foliar application. Soil commandos entomopathogenic nematode used for the soil application. Example is Tenernema species. Soil commandos are Heterorhabditis species. Application: green commandos apply to the leaves and above ground parts of the plant, applied to the soil. What do you mean by entomogenous nematode and entomophagous nematode? Entomogenous nematode, they are generated from the nematode. They feed and multiply on the host. But in case of entomophagous nematode, that nematode only feed on the host without reproducing inside it. Example for entomogenous, they are generated from the insect, that's why we are calling them as entomogenous. Examples Tinernema species, Heterorhabditis species. Entomophagous, they feed on the insect host, that's why we are calling them as entomophagous rhabditid species. Nematodes are mass cultured in vivo in the lab by using Lepidopteran larvae like Galeria melonella, that is waxworm larvae, Quercera cephalonica, rice moth larvae, and Spodoptera lichura. The National Center for Integrated Pest Management is situated in New Delhi. Thank you. Thank you all.
In this session, we are discussing terminologies related with the biopesticides. So, first terminology is the entomopathogen. Entomo mean insect, pathogen mean disease causing organism. So, entomopathogen is refers to any organism that causes disease in insect is the entomopathogen. This entomopathogen may be bacteria, fungi, nematode, a virus or protozoa. Right. So this microorganism specifically infect and harm insects. Specifically infect and harm insect and eventually help in managing their populations. Right. Come to the next slide. Then what is insect pathology? Study of insect diseases is the insect pathology. These diseases may be caused by virus, bacteria, protozoa, fungus, etc. Then, what is the difference between infectivity, infection? Infectivity, it is the spreading ability of the pathogen to the new host. The spreading ability of the, hello madam, the spreading ability of the pathogen to the new host and infect them is the infectivity. Then what is infection? Invasion and multiplication of pathogen in a host is the infection. Then what is pathogenicity? After infection, the ability of the pathogen to cause diseases in the host is the pathogenicity. Next, come to what is a virulence. It is the severity or degree of harm a pathogen can cause to a host is the virulence. For example, in 2021, the coronavirus is more powerful and it caused so many deaths. Therefore, it is highly virulent at that time. But now this is that coronavirus is low virulent. When we are saying the virulence of the protozoa is 90 days, virulence power mean it is effective for a period of 90 days and cause harm to their insect host. Virulence, it is the effectiveness period of any microorganism or pathogen. Next, come to the dosage. Dosage refers to the quantity or concentration of the infectious particle required to initiate a infection in a host organism is the dosage. See here dosage of NPV. It requires 1 into 10 to the power of 9 POBs per ml in order to cause infection to the insects. Therefore, here several examples are given for their dosages. Next, what is the sign, symptom, and syndrome? Physical or structural abnormalities in an organism or in an insect as a result of infection is the sign. The sign may include abnormalities like change in color or malformed appendages. When we come to the symptom, functional or behavioral abnormalities in an insect as a result of infection is known as symptom. Example for this is abnormal movement or there is no movement in the body part of the insects. Then what is syndrome? The collective name of sign and symptom is a syndrome. The collection of sign and symptom associated with a particular disease or abnormality is the syndrome. This syndrome, it is a Greek word, its meaning concurrence, means sign and symptom occurs at a time. Then what is the incubation period? The time period between infection and appearance of the symptom. This time period is known as incubation period. During this period, we can't see the symptoms or sign in an infected host. Course of infection. Course of infection refers to the progression of a disease, that is, how a disease develops over time within an infected individual is the course of infection. The stages of course of infection include exposure, incubation period, onset of symptoms, clinical phases, resolution or complications. Infection are of two types, that is acute infection and chronic infection. Acute infection occurs suddenly and lasts for a shorter period of time, results in the death of the organism. But chronic infection develops slowly and lasts for lifetime. Organism may or may not die. Then what is the latent infection or hidden or concealed infection? Following infection, some parasite may remain in latent or hidden state in the host tissues and may proliferate and produces diseases when the host resistance is lowered. Such infection is known as latent infection. Right? When resistance power of host is more,
then they are hidden or inactive. When the resistance power of the host decreases, they proliferate and show their reaction. Such infection is known as latent infection. Next, come to the zoonosis. Transmission of diseases from animal to the human is known as zoonosis. These diseases are generally transmitted via direct contact, aerosols, or bites. See here some example: brucellosis. It is come from cows and pigs. Anthrax from sheep. Plague from rodents, and rabies from bats and dogs. Then what is the epizootic and enzootic? Serious or outbreak level of the disease which occur over large area. For example, in 2020, that coronavirus occurred over large area in serious form. Such diseases are known as epizootic diseases. Serious or outbreak level of the disease over large area. Less serious or low level of the disease which occur in a particular area is the enzootic disease. Then what is diagnosis? The process of identifying a disease condition by examination or laboratory test is the diagnosis. Then what is etiology and epidemiology? The study of cause or origin of the disease is the etiology. Disease may be caused due to environmental factor, pathogens, or behavioral influences. So we are here studying the cause. The study of cause or origin of the disease is the etiology. Then what is epidemiology? The study of how the disease is spreading and how they can be prevented is the epidemiology. Then what is biopesticide? Living organism or their products used for the management of the pest is the biopesticides. These biopesticides still have limitations such as their broader effect on non-target organism and variable environmental impact. Broader effect means they may sometimes affect non-target organism also, and they also leaches into the soil, and they also affect on soil microorganism. So these are the limitations associated with the biopesticides. Over these biopesticides, the most accurate pesticides are the biorational pesticide. Biorational pesticides are specifically formulated, precise pest targeting, eco-friendly pesticides. See, for example, there one drop is given when we are using lure for helicoverpa, then it is attract only helicoverpa. It is do not impact any other organism. That's why it is specifically formulated and precisely pest targeting. So this is the advantage of biorational pesticide over biopesticide. Then what is botanical pesticides? Pest control substances which are derived from the plant are known as botanical pesticide. See here a list of various botanical pesticide is given, that is neem oil, pyrethrum, rotenone, sabadilla, nicotine and rania. So the ingredient or active component of neem oil is azadirachtin, nimbin, solanin. In case of pyrethrum, active ingredient is the pyrethrin. In case of rotenone is the active ingredient. In case of sabadilla, veretrine and alkaloid are the active ingredients. Then after, in case of tobacco, nicotine is the active ingredient. In case of raniodine is the active ingredient. All these things are covered in previous slide in tabular form. The last slide of this session is the classification of biopesticide. The US Environmental Protection Agency identified three main categories of the biopesticide. That is, one is microbial biopesticide, second one is the biochemical biopesticides, and third one is the plant incorporated protectants. So under microbial biopesticide, bacterial biopesticides are there, fungal biopesticide, nematode biopesticide, then after viral biopesticide, protozoan biopesticides are there. Under biochemical biopesticides, botanicals as well as semiochemicals are there. In case of plant incorporated protectant, that is cry protein, that is bt protein is incorporated into the cottons and brinjals, and some mRNAs, that is double stranded RNAs, also incorporated into the plant for managing the pest. When they feed on such plant, their development will be hampered. Like that, this plant incorporated plant also help in protecting our crop from the pest infestation. Thank you. That's all from my side. Any doubts?